condition called malignant peritoneal mesothelioma. I became interested in this many years ago because I was struck by the fact that in the oncology field there was really quite a nihilistic perspective on this particular condition. No one really had any effective treatments for it. We were interested in seeing if we could develop a strategy of using surgery as well as chemotherapy applied directly into the abdominal cavity to try and control the growth of mesothelioma within the abdomen. Mesothelioma is generally thought to be caused by asbestos exposure, although there are other risk factors associated with it. And there are two fundamental types, one that grows primarily in the chest cavity and one that grows in the abdomen. And my own area of interest has been in the abdominal form of this disease. It only affects several hundreds of people in the United States every year, but once it is diagnosed, it is really considered an incurable condition. What we found is that when people undergo surgery to remove as much of the mesothelioma as possible from the abdomen and then apply a high dose of intra-abdominal chemotherapy, that many individuals did quite well for a very long period of time, and we were really struck by these findings. At the same time, there was a group of individuals who did not do well. So my laboratory and I have been studying this condition, and over the years we have systematically collected tumors from individuals who have undergone treatment, and we've studied these in the laboratory. We've studied them using very sophisticated molecular techniques, and we have been able to find really quite remarkable insights into the nature of this cancer. When you study a tumor by looking at it under a microscope, it's like trying to understand the personality of a cancer, like people in a room being looked at from a plane flying miles in the sky. But when you use these very sophisticated molecular techniques, it's like parachuting out of the plane, walking into the room and staring the cancer right in the face. And by using these techniques, we've been able to look at the expression of thousands of genes and the proteins that they produce and the network of interactions that occur that provide them with a personality that really controls their growth and spread. And we've been able to find that really at a very molecular level that the growth of these tumors appears to be programmed into them from the very moment that they are diagnosed. We have found in all of these mesotheliomas that there are proteins that are present in very high abundance, which has never been appreciated before. And we hypothesized that these proteins are critical for the growth of these tumors, that the tumors are really addicted to this protein in order to survive. And by using techniques to eliminate the protein from the tumors, we've been able to show that they will not grow, either in a culture dish in the laboratory or in laboratory mice. And we are now very excited about our next phase of research, which will be to use drugs and agents that are available to us that we can try for patients with this condition in the clinical setting. We hope that this will help define a better, more effective treatment for individuals with this condition. Much of the work that has been done in this area has really been through the efforts of my very talented researcher, Dr. Shilu Varghese, who's really one of the brightest minds in cancer research, who I feel very fortunate to have working in my laboratory here at the Greenebaum Cancer Center. One of the things that he has pointed out to me is that this protein that we've identified in this fairly rare cancer, mesothelioma, is also abundant in very high levels in many other very commonly occurring cancers. So the discovery that we've made here in mesothelioma may actually have broad applicability to the treatment of many other commonly occurring cancers that we face in this country. We have found that there are compounds that are available for us to use in the clinical setting that can completely inhibit the function of these proteins to which the mesothelioma appears to be addicted, and we are